Welcome back! On the previous video, we discussed the pancreas anatomy, the contents of the pancreatic juice, and the function of the pancreatic digestive enzymes. First, let's start this video by discussing the phases of the pancreatic secretion. These are the same phases that are shared with the gastric phases of digestion. There are three phases: the cephalic phase, the gastric phase, and finally the intestinal phase. Let's start talking about the cephalic phase. The cephalic phase happens even before swallowing the food and is made up by various stimuli like the emotional state, the anticipation for the meal, visual, olfactory, and even auditory stimuli that are associated with a meal. These inputs will stimulate the parasympathetic nerve endings, which will increase the release of acetylcholine. This phase is responsible for 20% of the total pancreatic juice secretion, but it does not stimulate the flow of said secretion into the duodenum. Next is the gastric phase, which occurs by gastric distension, occurring during the gastric digestion of a meal. This continues the stimuli of the parasympathetic system and the acetylcholine release. This phase accounts to produce about 5 to 10% of the total pancreatic enzymes, but as with the previous phase, it does not stimulate the flow into the intestine. Finally, we have the intestinal phase, which happens when the chyme enters the duodenum. This phase is very important as it will acidify the pH of the duodenal lumen, which will stimulate a particular subset of cells called the S cells, which are responsible for the production of secretin. Secretin is a peptide hormone that is secreted by S cells when the pH of the duodenal lumen reaches 4.5 to 5 in the pH scale. Secretin is released directly into the bloodstream. This hormone will have a direct effect on the pancreatic secretions, increasing the bicarbonate ion secretions. First, the pancreas secretes bicarbonate ions in the form of sodium bicarbonate that joins together with the hydrochloric acid that comes from the stomach. This will react to form sodium chloride, which is a neutral salt, and carbonic acid, which will immediately dissociate into water and CO2. And since CO2 is a gas, it will easily diffuse to the bloodstream and be excreted via the lungs. This way, secretin and its neutralization serves various purposes. First is the protection of the duodenum. The intestinal mucosa does not have the capability of withstanding an acid pH coming from the stomach. Thus, the bicarbonate secretion prevents the formation of duodenal ulcers. It also provides an appropriate pH for an optimal functions of enzymes, as pancreatic enzymes need a pH of around 7 to 8 to function properly. Finally, since the acid pH is the main mechanism responsible for the secretin secretion, the neutralization will stop the release of secretin, or until another meal comes by. And finally, we have cholecystokinin. Cholecystokinin is a very important peptide hormone that is related to pancreatic secretion. This hormone is produced in the duodenum by a subset of cells called the I cells. The release of cholecystokinin is stimulated by the presence of small peptides, free amino acids, and free fatty acids in the chyme. Like secretin, it is released directly into the bloodstream, but instead of changing ion concentrations, it stimulates the secretion of much more pancreatic digestive enzymes in the pancreatic juice. It can also stimulate the parasympathetic vagal nerve endings to release even more acetylcholine and potentiate its effects on pancreatic secretion. Once digestion is completed and there are no more nutrients left in the duodenum, the stimulus stops to produce further cholecystokinin. Thanks for watching, and I hope this helps in your education.